Lipid metabolism. Lipids don't have clear definition, but it's a type of fat. Lipids are biologically significant and include neutral fats, phospholipids, cerebrosides, and sterols. First, let us see about neutral fats. Neutral fats, in particular, are fats that are described as neutral because they are uncharged and do not contain acidic or basic groups. It's nothing but neutral fats contains glycerol and fatty acids. Phospholipids. Phospholipids are a class of lipid whose molecule has a hydrophilic head containing a phosphate group and two hydrophobic tails derived from the fatty acids. Here, it has the attachment of glycerol plus phosphate group in head and two hydrophobic tail derived from the fatty acids, which has the character to repel against water. And cerebroside present maximum in the nerve cell membrane. And here is the structure of hydrophilic head and hydrophobic tail. Sterols. Sterols synthesized in guanine. It's about 30 to 50 percentage of dietary lipids are hydrolyzed. Therefore, an alcohol of guanine. Storage of fats. Mobile fats is in the form of minute droplets known as chylomicrons. Chylomicrons are large triglycerides, rich proteins produced in enterocytes from dietary lipids, namely fatty acids and cholesterol. Chylomicrons transport lipids absorbed from the intestine to adipose, cardiac, and skeletal muscle tissues, where their triglyceride compounds are hydrolyzed by the activity of the lipoprotein lipase, allowing the released free fatty acids to be absorbed by the tissues. All fat in the body does not come from dietary food, but it's readily synthesized from the excess of carbohydrates not stored as glycogen. Liver in fat metabolism. Liver is normally not an accumulator of fats as it is for carbohydrates. Liver contains about 3 to 8 percentage of fat content. The phospholipids and short chain fat acid absorbed via the portal blood are brought to the liver. It takes up free fatty acids from their systemic blood and then triglycerides from the chylomicrons. Now let us see about the steps of metabolism. Oxidation of fats. The oxidation of fatty acid is a stepwise process involving the cleavage of two carbon fragments starting from the carboxyl end of the chain. The fragmentation produces acyl CoA and the process by which it is formed is called beta oxidation of fatty acids. Activation. Before a fatty acid is oxidized, it must be activated by converting itself to an acyl CoA derivative with the help of activating enzyme. The enzymes catalyzing these reactions act according to the specificities upon short, medium, or long chain fatty acids. During this reaction, a molecule of ATP is consumed. First oxidation. The enzyme which involved in the first oxidation is acyl CoA dehydrogenase. And the product we get in the first oxidation is trans delta square enoyl CoA. And FAD has a special character which will remove the hydrogen. Second oxidation. During this process, two H atoms are removed, each from the beta carbon and hydroxyl radical by NAD. This resultant compound is called a ketosyl CoA derivative. Here we have the beta oxidation cycle. As we told earlier, the enzyme which involved in the first oxidation is acyl CoA dehydrogenase, and the end product we get is trans delta square enoyl CoA. And the enzyme involved in second oxidation is enoyl CoA hydratase, and NAD plus also removes the hydrogen, and the enzyme involved is L hydroxyl CoA dehydrogenase, and the enzyme thiolase gives at the end product as fatty acyl CoA. A palmitic acid molecule contains 16 carbons. Each acyl CoA molecule finally enters the Krebs cycle. The H atoms produced during the dehydrogenation process will undergo phosphorylation oxidation. Regulation of lipid metabolism. The metabolism of lipid is also influenced by the nervous system and hormones. Insulin, growth hormones, cortisol, reproductive hormones, and thyroxine are the hormones having their significant effects on lipid metabolism. The abnormalities due to defective metabolism is: first, we get obesity. It is only due to the ingestion of more food than required to meet the normal metabolic needs of the organism. In this, there is excessive deposition of fat in the depots with decreased mobilization. Second, Gaucher's disease. In this abnormality, cerebrocytes are increased in the brain, liver, and spleen. There is an imbalance between synthesis and the breakdown of this lipid. Cachexia is a disease, it has the opposite condition where the fat depots are very scanty. It shows very severe ill health. Thank you.